Good morning or good afternoon, everyone, and welcome to our webinar, Stem Cell-Derived Exosome Production in Stirred Tank Bioreactors. This webinar is part of our virtual event, Eppendorf Planet of Knowledge, here on LabRoots. Following the webinar, take your time and have a look at our event platform and discover more content and workflow solutions for your research. My name is David Solbach, the moderator of today's session, and now let me introduce to you my colleagues Aurélie and Jorge, who will present their work on exosome production in stirred tank bioreactors to you. And with this, I will hand over to Jorge.
Yeah, thank you, David. Hello, everyone, and welcome to today's webinar entitled Stem Cells-Derived Exosomes Production in Stirred Tank Bioreactors. This is the agenda we prepared for you today, and we divide in three main topics. First, the introduction, I will discuss about exosomes as a promising cell-free therapy tool. Then we will discuss about the challenges behind exosome production process. The second process, we will describe about process development in Dashbox mini-parallel bioreactor system. And then at the end, I will explain the exosome production on Siberia twin bioreactor control system. Yeah, as everybody knows, stem cell therapy, also known as regenerative medicine, is designed to repair damaged tissue in the human body by implanting new adult stem cells, modulating the immune system, and reducing inflammation. The premise of stem cell therapy is to help the body to heal itself, increasing the quality of life for patients. So you can see here more than 5,000 clinical trials have been used for different applications. The global stem cell market is very competitive nowadays, and it's estimated to reach around $146 million by 2021. This market is fueled to the therapeutic action of stem cells for several disease treatment and because of the stem cell line banking. In addition to that, the development of induced pluripotent stem cells, iPSC, also has contributed to the growth of this market due to the social ethical issues associated with embryonic stem cells. As you can see here in the picture of the musculoskeletal disorder segment dominating the global stem cell therapy, but unfortunately, clinical trials have made only a modest improvement in restoring normal functions to the regenerative tissues. In the earliest days, the mechanism of action of the cell therapy claimed that cells implanted should integrate in the site of injury, replace the damaged tissue, and improve tissue and organs. However, limitations associated with cell localization and cell survival in the target tissue works against tissue regeneration. But the real question here is, do we have the right tools to address the cell therapy limitation? Let's go through that. Extracellular vesicles, including exosomes, are one of the most important paracrine effectors involved in the intercellular communication. They were discovered more than 30 years ago, and they were considered as cellular waste disposals. Think of an exosome as an envelope with instruction inside, like one cell mailing a letter to another one, telling it what to do. In this case, the envelope, that is the exosome cargo, contain microRNA, non-coding RNA, and a lot of proteins. They are released from different types, such as T and B cells, mesenchymal stem cells, tumor cells. As I mentioned before, they mediate inter and intracellular communication when they are secreted in the bloodstream, and also they modulate the immunoregulatory processes, set up tumor escape mechanisms, and mediate regenerative or the regenerative process. They have also a round-shaped morphology, and they are present in the variety of body fluids such as blood, urine, amniotic fluid, synovial fluid, etc. The cell secretome consists of extracellular vesicles, specifically exosome, microvesicles, membrane particles, peptide, and small proteins. So you can see here, we show this picture because we have a comparison between them, but exosomes are lipid bilayers vesicle with a diameter ranging between 50 and 100 nanometers, and also they can be easily confirmed by first markers such as CD9, CD63, CD81 of the transplants. Yeah, but not all the mesenchyma derived stem cell derived exosomes are similar. The mesenchyma stem cells are present in different adult tissues such as adipose tissue, bone marrow tissue, umbilical cord, as well as iPSC and embryonic stem cells. And depending on the source, the exosome yield is completely different. So they offer several advantages compared to the cell-based approaches, including sectional stability and biocompatibility. Moreover, they can be delivered to target tissue easier than the whole cells, and they can migrate across the blood-brain barrier. According to the PubMed, and you can see here in the slide, the number of citations has grown more than 10 times in the last decade, so they have gained in importance. Today, this new cell-free therapy has had great impact in various fields of application, reaching 192 clinical trials worldwide related to exosomes that are listed in clinicaltrials.gov. So they have been used as drug delivery systems, as tumor biomarkers, and mesenchyma stem cell derived exosomes also have great potential in bone and cartilage regeneration. These figures show the biotherapeutic particle history since 2006. So as you can see here, in 2006 was approved the first cancer vaccine. Later in 2012 was approved the first gene therapy, and in 2015 was approved the first oncologic biotherapy products. So, and now I will let my colleague Aurélie to describe the challenge behind exosome production process.
Thank you, Jorge. Now you have a clear view of what's an exosomes and their growing interest in the context of cell-free therapy. Nevertheless, the exosome production process is still challenging. And the preclinical and clinical development of the exosome technology as a cell-free therapy or drug delivery platforms require large quantity of exosomes. It's really difficult to define a precise range regarding the optimal therapeutic dose. Indeed, this therapeutic dose will depend on the specific purpose of the study and on the way the exosomes are quantified, but you have here an example of the huge quantity of exosome which is required to be administered to an animal to achieve biological outcomes. So the mass production of exosomes is then a requirement to generate clinically relevant amount of exosomes. However, the mass production of exosomes presents several challenges. Indeed, you have to reach a delicate equilibrium between the quantity and the quality, and the final product must be pure, but also keep its biological property. The manufacture process you have to use should be scalable, of course, but also have to be controlled, standardized in order to improve reproducibility. And finally, this process should be in accordance with the appropriate regulation requirement. So for the moment, the most current methods used to enrich, collect, and isolate exosomes are not fitting with those aspects. The exosome production workflow is a multi-step process and could be divided in three major steps. The upstream process here first, during which the cells used as exosome sources are expanded, and also during which the exosomes are enriched in the cell-conditioned medium. The second step is the downstream process, consisting in the isolation of the secreted exosomes from other elements contained in the conditioned medium. And finally, the quality control, which is based on the exosome characterization. And for all those steps could be a source of variability and a challenge for the researcher. And for example, for the upstream process, the question of the researcher could be which exosome source to select, or how to ensure an efficient cell proliferation and exosome secretion, how to deal and how to proceed to the in-process control. So before to go more in detail with the upstream process, which is the topic of today, I would like to briefly describe you some challenging aspect and key message associated with the downstream process and the quality control. There is a large variety of techniques which could be used to isolate exosomes. Historical gold standard methods are based on ultracentrifugation with two main variations, either differential pelleting or using density gradient. But in a manufacturer point of view, the ultracentrifugation-based methods are present some limitation, especially because they are difficult to scale up. The process is often long, multi-step, and require high level of skill and expensive equipment. So it exists a lot of different other techniques to isolate exosomes, such as size exclusion chromatography, filtration-based method, precipitation, or immunoaffinity capture. Those methods present a better scalability, but all those methods present advantage or limitation, of course. Here is some important message to consider for exosome isolation strategy and selection, especially in the transition from a bench scale to a clinical production of exosomes. So first of all, a very important message is that for the moment there is really a lack of standardization for the exosome isolation strategy. Nevertheless, the isolation strategy you choose will greatly impact the final product. So it's really important to keep this in mind. Another point is that often researchers will use combined techniques in order to improve the purity of the final product with a balance between recovery and specificity. And the choice of the technique, the choice of the strategy to combine together to isolate your exosomes will really depend on the final purpose. So that's once again the question of the balance between the recovery and the specificity. In a context of mass production of exosomes in a therapeutic purpose, the crucial aspects to keep in mind are, of course, scalability, respect of regulation, potency maintenance of the final product, process control, but also specificity and high efficiency in terms of yield, cost, and time. So it's really challenging. Regarding the exosome characterization, there is also a very large variety of methods used, making result and analysis really difficult to compare. Between study from a study to another. So some methods are based on physical properties, others are based on the presence of specific markers. And recent review also highlight emerging techniques. But once again, there is some challenge, especially the standardization, the specificity, the sensitivity, and the throughput of those methods. And now we will focus on the first step of the workflow, so the upstream process resulting in an efficient exosome secretion. We would like to highlight key factors to address to improve and warrant a successful and reproducible upstream process. So as Jorge mentioned in the beginning of this introduction, not all exosomes are the same. Not all MSC-derived exosomes are similar. It's really important to keep in mind that the cell culture parameters used to improve and to process the cell expansion will largely impact the quantity and the quality of the secreted product and the secreted exosomes. So any change in the cellular phenotype due, for example, to the adhesion process, the proliferation process, or to any potential cellular stress occurring during this phase of the culture could generate unwanted alteration in the composition and the function of all those exosomes, as those two very interesting articles describe. So, of course, you could consider different parameters which could impact. It could be the culture medium, the culture support, but also other physicochemical parameters. Regarding the culture medium, the idea is to push an efficient and robust cell proliferation. Of course, to do that, we usually use growth factors, generally supplement in the medium as a fetal bovine serum. but nevertheless, those animal-derived supplements contain exogenous exosomes, which could cause contamination in the final product. So exosome collection could be then consequently performed differently, either using culture medium supplemented with exosome-depleted FBS in order to reduce the potential contamination. Take care that it's not a free of exosome medium, but exosome depleted. It will just reduce the contamination. Another way is to use a two-step process with the first phase, which ensures the cell expansion in a classical culture medium. As soon as you have obtained an optimal cell density, you will completely change the medium for an exosome collection medium, which is free of exogenous medium. In this medium, the cell will decrease the proliferation, but you will use this medium for a reduced period of time just to collect the exosomes. Some commercial versions of those medium exist and are supplemented to increase the exosome secretion. Another important parameter is the culture support, especially in a scale-up process. So conventional 2D adherent cell culture support, such as flask or multi-layer flask, are limited in terms of gross surface area per volumes. So usually we go in the direction of the 3D culture, especially based on the use of macrocarriers to increase the cell density using a bioreactor. And this culture in 3D could offer not only the advantage to be scalable, but also the advantage to be more physiological for the cells. And different papers have already described the fact that the use of a 3D culture could improve the yield and the functionality of the secreted exosome. In the transition for the 3D culture, there is a lot of parameters that you have to optimize in order to ensure a robust attachment and an efficient start of proliferation. Amongst those parameters, you could, of course, select the most appropriated type of carrier. The initial cell seeding density could also be optimized as the cell carrier ratio, and you could also optimize the first period of time in order to improve the adhesion with a reduced agitation, for example, or using a reduced volume. Finally, there is a lot of different physicochemical parameters which could impact your culture, and you have to take care to the pH, the oxygen tension, the temperature maintenance, the agitation, and of course the feeding strategy you will use. As an example here, in order to be more physiological, you could be interested to reduce the tension of oxygen in order to be more physiological, mimicking the in vivo niche of the stem cells. And all those parameters should be stable and more, sorry, could you mute? Thank you. In this way, the use of stirred tank bioreactors could be very interesting and offer you a lot of advantage. Those bioreactors allow you to process, to monitor and control your process during all the entire process. You could really control and find the ideal condition, maintain those conditions during all the process, and nevertheless, moreover, they propose a scalability option with similar vessel geometry and capability at different size. So it's very interesting in this way. As a conclusion of this introduction, we would like to insist on the importance of the standardization of the entire process through all this process, from the cell culture to the final characterization. And it's also very important to really describe in detail the material and method parts of the publication in order to be really reproducible and to reproduce exactly the same condition. In the next part of the presentation, we will introduce you the use of the Dashbox mini parallel bioreactor system to develop an exosome production process in a small-scale tire tank bioreactor and optimize the culture condition before a potential scaling up. So this first slide describes here the experimental strategy used for the upstream process. So MSCs derived from adipose tissues have been selected as an exosome source. After thawing, the cells will recover a few days in a flask. The appropriate amount of cells have been seeded on microcarriers in a BioBlue single-use vessels. And the cell growth is then monitored during 5 days by the Dashbox systems. Growth factors are added 3 days post-seeding in order to ensure proliferation. Regular monitoring of the cell adhesion, but also the cell viability, is performed during the expansion phase. After 5 days of cell growth, the culture medium has been completely changed for a collection medium, which is free of exogenous exosomes. And the exosome enrichment occurred during 2 additional days at the end before harvesting and performing downstream process. Here are the main process parameters used during this upstream process. You have here the type of carriers and the quantity we have used, the inoculation cell density and the working volumes. Importantly, we have note here the cell carrier ratio and the dissolved oxygen percentage, which correspond to a pressure of oxygen of 8%, mimicking the in vivo environment. We have also the pH and also information about different parameters such as the agitation. We have the agitation for the first 4 hours, and then the agitation was set at 100 RPM during all the process. Here are the first results. Cell viability was determined regularly during the process by staining with a fluorescent calcine AM probe and visualized under a fluorescent microscope. As you can see, through the 5 days post-seeding, cells are growing progressively and form progressively small aggregates and 3D structure. The proliferation has also been evaluated through cell counting performed at different time points during the process. So small samples of the cell suspension are taken from bioreactors and cells are detached with trypsines and counted with a ViCell automated cell counter. The results obtained confirm the efficient proliferation with a very short lag phase at the beginning, and the final density is noted here. You have here the complete the total number of cells in the vessels, which and the fold increase we have obtained after 5 days. In the meantime, we have also checked the concentration in glucose and concentration in lactate, which are is secreted in the culture medium, and those parameters also confirm that the proliferation is efficient. Now, here is the experimental process used for the downstream part. We have collected, of course, the cell-conditioned medium, and this medium has been centrifuged, filtrate, and concentrate using those parameters. A small sample is safe to perform particle size distribution analysis, and the rest of the volumes is loaded on a size exclusion chromatography column. We collect at the end 22 fractions, and the total protein content is also evaluated during all this process. At the end, every fraction is concentrated before to be submitted to exosome characterization, and in our case, we have selected to study the presence of the CD63 protein, a transmembrane tetraspanin protein, which is specific of exosomes, through thanks to an ELISA approach, but also with a Western blot approach. Negative control, it's really important, was also performed in parallel. It consists in a non-conditioned medium, so it's the collection medium without with no contact with the cells, and this negative control has followed exactly the same procedure, the same process than our conditioned medium. Here are the results obtained with the elute fraction using the ELISA and the total amount count. So in blue, you have the total amount evolution in all those fractions. and you can see that there is a peak here on the fraction 20, 21, and 22. In parallel, we have also the result from the ELISA assay, and here you have seen that we have a peak on the top of the fraction 18 and 19, with a maximum of quantity in the fraction 19 corresponding to 9 to 10 exosome quantity. We confirm those results with a Western blot analysis, and here again we have an increase of the CD63 protein in the fraction from 16 to 20, with a peak at the fraction 18 and 19, 17 till to 19. Finally, we evaluate the size distribution of the samples we have collected through two different methods, the dynamic light scattering and the centrifugal liquid sedimentation analysis. Once those two methods confirm that we have a population or a bimodal distribution with two nearby population at size ranging in the range of the exosomes. So to conclude this part of the presentation, we can say that the Dashbox mini reactors is a perfect tool to test and optimize the process at small scale. You can really compare and test the influence of different culture parameters during all the process. It's really easy to compare. You could monitor every physical-chemical parameters in a very stable environment in order to define reproducible parameters to reproduce at the end. So now I would like to let Jorge present another and different strategy in order to produce exosome in the Siberia.
Thank you, Aurélie. Yeah. A different strategy was followed in this study. In this case, as you can see here, we use BioBlue single-use vessels as bioreactor for induced pluripotent stem cells, derived mesenchyma stem cells, exosomes, production, isolation, and characterization. We employ the Siberia twin bioreactor control system as a control device and collagen-coated microcarriers as cell culture support. The main aim in this study was the production of exosomes using 1 liter scale as a model for large-scale production in bioreactors. To demonstrate the ability of iPSC-derived mesenchyma stem cells to secrete exosomes, we performed two medium exchange experiments in 4 steps, as the picture showed. Yeah, in the step number 1, details the inoculation preparation. We use T-flask culture conditions for the initial cell expansion and hyperflask in the third process to achieve the appropriate inoculation cell density. Then, the stemness capacity of iPSC-derived mesenchyma stem cells was analyzed by flow cytometry, noting that they display typical phenotype of mesenchyma stem cells, positive for CD90 and CD29 typical mesenchyma stem cells market, and negative for hematopoietic markers such as CD34, and then for monocytes CD11B. So then iPS-derived mesenchyma stem cells were transferred into glass bottle containing the collagen-coated microcarriers and cell culture medium. In this case, we use 2 different kind of media, NF12 and ATCC complete medium. Then we kept the bottles in the incubator for 2 hours under certain conditions, as you can see here. Yeah. On the other hand, prior to the preparation of the BioBlue 1C single-use vessels, we perform a calibration of the pH sensor, and then we outfitted each BioBlue, among others, with a deep tube along with a compressed probe adapter, both inserted in the storage PG 13.5 port under aseptic conditions. I would like to highlight here that this big tube was used to do the medium exchange. Following the incubation, Following the incubation, 300 milliliters of cells on microcarriers were transferred into the BioBlue 1C bioreactor already filled with 700 milliliters of medium, reaching the working volume of 1 liter. Yeah. After day 5 of cell culture, we performed the medium exchange every 2 days until day 9, and then daily basis as required. But how we did that? So we stopped the agitation and gas flow for 5 minutes, allowing the microcarriers to settle in the bottom of the BioBlue 1C and exchange 10% of the medium using that big tube to remove the cell culture medium from the surface and using the feeding port to add fresh medium. Yeah. After day 5, 8, and 11, day 5, 8, 11, and 14 days of culture, we collect 50 milliliters of iPS-derived mesenchymal stem cells along with microcarriers and medium using a container bag with line set. So we transfer that 50 milliliters into 50 milliliter conical tube along with iPSC-derived mesenchymal stem cells and microcarriers in the bottom. So then we remove that supernatant and added 50 milliliters more medium using the same composition, but in this case replacing the FPS with 2% of exosome depleted FPS, and then we transfer into the shaking flask. So finally, we incubate that iPSC-derived mesenchymal stem cells and microcarriers for 48 hours under the same conditions. So then 48 hours after each collection day, as you can see here in this slide, we remove the supernatant and then we perform the isolation purification of exosomes according to the ExoQuiz protocol with some modifications you can see here. So we collected samples every day in the course of the first experiment and every 2 days in the second experiment from bioreactors to determine the cell viability, cell density, and metabolite concentration. So 3 milliliters were collected for analysis. Then these volume were transferred into the conical tube. So we wash with PBS and then we trypsinize the cells in order to detach the cells from the microcarriers and do the analysis. So later on, the cells and microcarriers were filtered through a 40-micron strainer in order to determine the cell viability and density. As you can see here in the picture, after the cell expansion, the cell count was erratic, especially after each collection day. So we attribute this to the low inoculation density and also because we use in the first experiment the DMN F12 cell culture medium, whose composition probably won't be suboptimal for the iPSC-derived mesenchymal stem cells expansion. Also, we performed a second experiment to increase the iPSC-derived mesenchymal stem cells inoculation density and the exosome production yield for each sample. So to achieve that, we changed the cell culture medium. In this case, we use ATC complete medium, and also we increase the inoculation cell density. Finally, we kept the rest of the conditions in the assays of the first experiment. As you can see in picture A, we observe an initial lag phase 24 hours after the inoculation, followed by the steady increase of cell growth between day 1 and day 9. So then the culture reached the stationary phase on day 9, followed by a subsequent decrease in the cell density until day 15. I have to add here that we observe a substantial microcarrier aggregation later in the course of the run. Therefore, we base the cell count in the later stage not only to the floating microcarriers, to the floating microcarriers. So probably the average of the cell expansion in the base must be higher. In addition, so we determine the consumption of glucose and production of lactate and ammonia. As you can see here in the pictures, exosomes are highly enriched in protein in which the transponins membrane, as I mentioned before, CD63, 9, 81, and 82 play a key role in cell invasion, penetration, and fusion events. So we use a direct enzyme link non-sorbent assays, ELISA, specifically ELISA Ultra C63 kit, in order to quantify the exosome abundance. The results show that the number of iPSC-derived mesenchymal stem cells exosomes constantly increased from day 5 to day 16. So in addition, we found a direct correlation between cell density and secreted exosomes during the whole experiment. To study the three-dimensional cell morphology of poly-encoded microcarriers, we collect samples at day 5 and 9, as you can see here in the picture, and then visualize them through the bright field images. So we evaluate the morphology of human iPS-derived mesenchymal stem cells, demonstrating that, as you can see in the picture on the left side, at the early stage of the cell profile, a few cells attach on the microcarrier surface and progressively form inter-microcarrier cellular bridge and aggregates, as you can see, day 9. So it's well known that a 3D cellular environment allows the cells to mimic the in vivo cellular behavior involving cell-cell and cell-extracellular matrix interaction and promoting cell signaling and proliferation. So it has recently been reported that although the mechanism of exosome biogenesis is still not well understood, findings suggest that the exosome secretion and molecular cargo can be altered by the cell's microenvironment. So it's important to study morphology. So finally, I would like to add that these various twin experiments are preliminary studies and have not yet been optimized to obtain the maximum exosome production levels. However, our observations can serve as a guideline for further improvement in mesenchymal stem cell-derived exosome isolation, purification, and scale-up protocols. Thank you very much.
Thank you, Aurélie and Jorge, for the presentation. For everyone, if you haven't done yet, there's still time to submit your questions via the Q&A window. To give you a little bit time to do this, I would like to show you in more detail 3 of our products that we used for these studies. First, there's the CellExpert CO2 incubator. You do not need to worry anymore to place your plates always at the exact same position, because with our incubator we verified the uniform temperature throughout the whole system at 27 different spots. So we really ensure that there's a homogeneous growth condition. The second one Aurélie talked quite extensive about was the Dashbox mini bioreactor system. This is a perfect system that simplifies your process development. It's a 4-fold unit, but it can be extended up to 24 bioreactors. And together with the possibility to easily integrate third-party analyzers and the combination with our powerful Dashbox software suite, the Dashbox is the perfect tool for the efficient process development. And last, the Siberia twin, our newest bioprocess family member, from the experiments and the results that Jorge did present. The twin controller already supports 19 different vessel variants, and there will be even more to come in the future. The innovative pay-drawer concept of the system allows you to easily adjust it for changing requirements in the lab. If you want to have more information on this product, you just visit our website eppendorf.com/Siberia. I see now that there are a couple of questions. So I will start reading with the first one, and this is going to Jorge, if I see it correct. The question is, how do you ensure that you do not have exosomes from the growth media in your experiments?
Oh, thank you, David. This is a good question. Well, a critical aspect here has to be considered before exosome production. And essentially, is the cell culture. So as you know, adherent cells require medium supplemented with CD of FBS in order to attach it in any surface. And we had before, when we were designing this experiment, we certainly performed several preliminary studies to establish the best conditions. So one of them was determine the medium composition. So we use medium with our FBS and medium with different exosome depleted FBS. Finally, we found that 2% exosome depleted FBS was sufficient to maintain cell viability in the ground. But this is one thing that we have to see in the future. We need more development in cell corrosion in order to use it for this kind of applications.
Okay, thank you. The second question is focusing, is for Aurélie. Why are mesenchymal stem cells a common cell type used in clinical trials to generate extracellular vesicles?
Since few years, we know that the MSC secretome, paracrine factors secreted by MSCs, are described to play a very important role and have biological outcomes. It could be cytokine, chemokines, growth factors, but also EVs, including exosomes. And EV derived from mesenchymal stem cells have been described to have a different potential, interesting effect, such as increasing angiogenesis and wound repair, reduce myocardial ischemia, reperfusion injury, promote a large variety of tissue regeneration. So that could be the reason why the MSC is common.
Thank you, Aurélie. The next question is for Jorge again. Have you compared the results obtained by ELISA with other quantification methods?
Yes, thank you, David. So we used 3 different methods by the exosomes in the preliminary study. We used the micro CA putting aside to the total concentration and after conversion. And then we used human CD66 detection kit, exosome quantification using the flow cytometry. And as I mentioned in the presentation, I used was the ELISA at 25, because this specific assay gave us better results and logical exosome production trend.
Okay, I hope everyone could understand this and the audio connection is not too bad. The next question is also for Jorge. Why did you use iPSC-derived MSCs and no other MS cell sources?
Okay, that's a good question. So, as I mentioned before, one of the preliminary studies that we did was the selection of the cell source. So we did 3 studies in parallel, using adipose-derived mesenchymal stem cells, bone marrow, stromal cells, and iPSC-derived mesenchymal stem cells. But what we saw in this experiment is that using the iPSC, I was able to get the highest yield of exosome under our specific conditions. So, but it's good that this is a good example for our customers. So we were not pursuing a particular cell source. We were able to use one and show the customer that with our conditions they can get good amount of exosomes, and also they can follow our protocols.
Okay, great. Is there a solution to just harvest the supernatant with the exosomes? Aurélie, that one is for you.
Okay. I would say that usually for the secrete cell-derived product, we could use fibrous cell disc and packed bed bioreactors, which offer a nice opportunity to make easier the collection of the final product. But with exosomes, there is some difficulties related to the medium composition and the presence of the exogenous exosomes in the medium. as we have mentioned in the first question. And so, as previously discussed, there is no perfect composition for the moment to promote in the meantime the cell proliferation very efficiently without exogenous EV contamination. So we have to switch the medium for EV collection medium, which is not so easy with the packed bed bioreactors.
And which isolation system do you see best suited for the future to scale up the process?
If I could continue, perhaps I could say
Yes,
that
please.
currently there is no perfect and standard isolation methods. Usually researchers use a combination of two techniques to improve the purity and reach the balance between specificity and efficiency. An interesting combination in terms of scale-ups could be the use of the tangential flow filtration first, followed by a more specific affinity capture step.
Yeah, if I can say something, that there's no standardized method that we can use right now in the market. So the best thing that we can do is to combine both in order to get a high pure cell population of exosomes.
Okay, thank you. The next question, I guess both of you can answer or have a different answer. It's which microcarrier did you use for your experiments?
Yeah, perhaps I can start. It exists really a large variety of different carriers on the market, and on the publication there is no a perfect solution for MSC, so there is large different possibilities. Some carriers could be macroporous, microporous, non-porous. Some present some surface coating, such as collagen or Synthemax coated carrier to improve the cell adhesion. So it's not so easy to select. So I think it depends on the cell type you use, but also on the medium composition. For example, when you use synthetic medium or medium which are reduced in FBS, you could use surface-coated carriers in order to improve the cell initial adhesion on the carriers. So that's what we have done for my part of the experiment. We have used the Synthemax-coated carriers.
Yeah, so I use the collagen coat carrier. So they're good cell support, especially at the beginning of the experiment when we need that the cells attach as soon as possible over the surface and avoid lag in our culture. So and with the collagen coat carriers are good microcarriers to address and to avoid certain limitation of cell attachment at the beginning of the experiment.
Okay. At the beginning of the presentation, you mentioned the increasing interest in clinical treatments. Jorge, can you give an example of exosome used in clinical treatment?
Well, when I was, of course, my preliminary study, the study that have been done, and then preparing this webinar, so I have been reviewing, of course, a lot of literature, but then I found, right now, as everybody saw in the presentation, that there are many products in the market and many clinical trials going on right now. But I would like to highlight one paper. The paper is from Professor Giebel and collaborator from the University of Essen in Germany. And they use mesenchymal stem cells derived exosome as a novel tool to treat therapy refractory graft versus host tissue, because mesenchymal stem cells are considered immunomodulating cells. I mean, this is for the people who are not very familiar with that, is when you implant donor tissue from one person, and then the host tissue reject this allogenic cell that you are implanting in this patient. So normally, what people use is corticoids in order to reduce inflammation, allow the immune system to accept the new cells. But basically, they use here exosomes with good results. I mean, if people want to read more information about that, I would be happy to share the paper. But basically, at the end, they found that they reduce the collateral side reactions due to that disease. And yeah, they explained that they did that in one patient, and they found good. They conclude that the exosomes may be a great potential to reduce inflammation associated disease. I think this is one example, but again, right now we have many options in the market, many clinical trials going on. This is a new field, and I have hope that many limitations associated with the cell therapy can be addressed using the exosomes or cell-free therapy, as people know this field right now.
There are really a lot of questions on the list, but I think we have time for one last question. Aurélie, I will give this one to you. How to reduce the lag phase often observed after seeding in a bioreactor?
Yeah, it's a good question, especially for people which are perhaps afraid by the transition from 2D to 3D culture. So of course the quality of the initial cells is very important. You have, of course, to use cells very young. The successive passage, especially for MSCs, could induce progressive senescence and change in the phenotype and decrease in the proliferation rate. The carrier selection is also important. We have already discussed this point, but also other parameters like the initial cell seeding density and the so-called cell-to-bead ratio. So it's really important to optimize those parameters with your system. Usually we use a ratio of 3 to 5 cells per bead. It's recommended, but you have to optimize this case by case and test with your cells and your medium. Another recommendation could be to really stop the agitation during the first hours just after the seeding of the cells, and work also with the reduced working volumes during this short period. It could help the adhesion of the cells to the carriers, and then a better start of the proliferation, a faster start. This is an interesting parameters. Yeah.
Okay, thank you both for all the detailed answers. For all the questions that we didn't answer yet, we will get in contact with you soon and write you an email with an answer to your questions. Thank you all for listening and enjoy the rest of the day. Bye.
Bye.
Bye.